It's a great honor for me to be recognized and by the MDS awarding me the David Marsden Lecture Award. I actually have met David Marsden, and in retrospect, it's a privilege to have met him because he was such a great giant in movement disorder. However, the time when I met him and the topic of alpha-synuclein in the pathogenesis of Parkinson's disease was just emerging. And so this was in the 90s. And but for the last 25 years, a lot have happened. And I think that there's just really a lot we know about alpha-synuclein. And hopefully, and the information that I share with you today will already, I think, in the process, but eventually, hopefully, will turn into biomarkers for diagnosing and movement disorder of Parkinson's disease, and also for treatment. And so, as the title of my talks, as transmission of misfolded alpha-synuclein protein in Lewy body diseases, and it really summarized some of the work that I've done in the last dozen or so years. And so basically, at the beginning, it's just some background slide, just showing you about the pathology. And let me say, I think there's one slide before this. It's not really responding, but never mind. So basically, these are the different pathology that you find in the brains of neurodegenerative disease patients. You're very familiar in Alzheimer's disease, beta amyloid plaque and neurofibrillary tangles. But in Parkinson's disease, it's this protein called alpha-synuclein. And they're the building block of these inclusions that was coined Lewy body by Dr. Lewy and a long time ago. And so basically here, these describe the progression of alpha-synuclein as a disease, as Parkinson's disease or movement disorder progresses. And this was described by Dr. Heiko Brack and his colleagues summarized in this review, and by Matthias Jucker and Walker. And so you can see that the Lewy bodies start really in the lower brainstem, and then they start moving and upwards, and then into the substantia nigra and all that, and midbrain, and then eventually spread to the entire brain. So basically, at the end stage of this disease, then you see a lot of alpha-synuclein pathology in the brain. And so, and basically, once you identify the protein that seem to form inclusions, then you want to identify the neurodegenerative diseases that have the pathology. So in addition to Parkinson's disease, and there are Parkinson's disease with dementia, or some people call it dementia with Lewy bodies, and also multisystem atrophy and Alzheimer's disease all have alpha-synuclein pathology. But multisystem atrophy is a little bit different in the sense that the Lewy body pathology are found in oligodendrocyte and not in neurons, whereas all the other neurodegenerative disease with alpha-synuclein pathology, the pathology are in neurons. But believe it or not, Alzheimer's disease is the most common and alpha-synucleinopathy or disease with synuclein pathology. So why is that? Because there's so many more Alzheimer's patients than Parkinson patients. Even though that you all hear this, you're treating clearly patients, but there are 5 times more Alzheimer's patients than Parkinson's patients. But at end stage of Alzheimer's patients, often they develop and movement disorder. They have Lewy body in their brain. So basically, the reason why this is important to me is that, if you see the rest of my talk, is that I really want to understand and define Lewy body and study how they eventually cause a disease. And so based on what I just said to you, that they're in this pathology, and they also seem to spread over time, and then I mentioned that they different and clinical phenotype with alpha-synuclein pathology. So that allowed me to put up 2 different working hypotheses. The first is the transmission hypothesis, and that is that these misfolded protein and can they spread, they can initiate and induce a cell-to-cell spread of Lewy body? And we want to demonstrate it in mouse model because that's one way to demonstrate that this is maybe happening in people as well. And the second hypothesis, and that I want to address, is the strain hypothesis. That is because there are some patients with MSA, some patients with Parkinson's, some patients with Parkinson's with dementia, and so on. So that do they represent different strains that could explain the clinical diversity? And so the way that we did those experiments, all these initial experiments were done by Calvin Look, and he's going to chair a session later on. And so, and this was in 2012, I guess, and so 10 or 12. And so what we did was that we just generated preformed fibrils. So you can actually generate recombinant alpha-synuclein in bacteria. And then you can basically, under different conditions, you can shake them and whatnot, and make them assemble into amyloid fibrils, which we call preformed fibrils, or PFFs. And at that time, we actually took a lesson from the prion field. And because basically, at that time, most people that work on alpha-synuclein, and they work on human alpha-synuclein, but in the prion field, they demonstrated their species specificity. So in other words, that perhaps then, the human protein may not work as well in a mouse model, and vice versa. So, so we decided to generate some mouse alpha-synuclein because we want to be sure that we don't have that that barrier, that species barrier. And so Calvin generated these mouse alpha-synuclein preformed fibrils, and then we wanted to mimic to see whether or not we can induce pathology and cause neurodegeneration. So we injected them into dorsal striatum, and then we basically just inject some PFF preformed fibrils into the substantia nigra, and then we just wait for different lengths of time, and you can see here, for example, even at 30 days. You're beginning to see some pathology forming in the brain of these animals. And these animals are wild-type animals. They're not transgenic animals that overexpress alpha-synuclein. They're just plain old regular mice. And so you can see they already developed these phosphorylated alpha-synuclein pathology at 30 days. And when you follow them longer, and this is in G, basically describe the spread of the pathology over time. And these red dots are the neurons that have alpha-synuclein pathology. This is where we injected. And then you can clearly see that the pathology increased over time, so such that by 6 months, 180 days, you can see a thin amount of pathology throughout the brain. And in the amygdala, in the olfactory bulb, in the striatum, and so on, and cortex. And also what is really gratifying is that we actually saw some synuclein pathology, phosphorylated alpha-synuclein, in the substantia nigra pars compacta. And as some of you probably remembered, that people have tried to do that, to get animal models to develop and Lewy bodies, but they just don't. You can generate MPTP models and so on. They just don't develop Lewy body, particularly in the substantia nigra pars compacta neurons. So this is very gratifying. And so basically, as I said, we follow them over time, and then we basically show that there's progressive disappearance of dopaminergic cells. So these brown stain that you see right here, and they're the ipsilateral substantia nigra. This is the side where we injected and mouse preformed fibrils and into the brain. And then you see that by 6 months, 180 days, there are actually much fewer brown cells compared to 30 days. And in fact, what is really convenient is that in the mouse, the left side and the right side of the nigra don't talk to each other. So you can actually use the other side, the contralateral side, as the control. You can see that these cells are not lost. They're all there, the dopaminergic cells. Whereas the other side, you see that there are much fewer cells. They're quantified here and over time. And so what also is really important is that as the dopaminergic cells die, what we found was that they have movement disorder. They have behavioral phenotype. They have weakness of their hind limb, for example. You see that in this wild hang behavioral test, and by about 6 months or so, they hardly can hang on to the cage. So this is, and it's also nice to see that because of the fact that we injected into the brain and there's no cognitive impairment, it's only movement disorder. So basically, we think that in this model, we have recapitulated and movement disorder. It's not, okay, all right. So in summary of this part of my talk, and this alpha-synuclein preformed fibers initiate conversion of endogenous alpha-synuclein and then accumulation into Lewy body, Lewy neurite inclusions in animal models. And that often inclusions drive the selective loss of substantial dopaminergic neurons, resulted in behavior impairment, reminiscent of human Parkinson's disease. So basically, conceptually, this is what I just talked about. And you have cells that have a misfolded protein, in this case, alpha-synuclein. And so it's misfolded, you can see, and that I illustrated here. And then, in order for it to spread, it has to somehow get out of the cell and be able to affect either neighboring cells or cells they communicate with. And then once they get inside, they corrupt the endogenous protein to adopt the abnormal conformation and then elongate it. And so basically, there are many different ways to target misfolded alpha-synuclein protein. You can block the release of the misfolded protein, and or you can actually block the uptake into the recipient cells or the cells that they talk to, or you can actually prevent seeding once it gets inside the cell, or you can actually promote degradation. And so there are different ways to come up with therapy using the scheme that I just described. So what I'd like to do is move on to tell you something about the strain hypothesis. So the idea would be that the pathological alpha-synuclein strain exists in alpha-synucleinopathies that potentially explain clinical diversity. So the concept is that their strain means that they have perhaps different conformation. And this is the hypothesis. We think that they have different conformation that would define the strain. You can see here, they have different shapes, for example. Okay, but then the normal, this is synuclein, and tau is the same thing too. So, but the normal synuclein basically can be, recruited and by the different conformation of strain or the different strain and seeds, and then elongated. And then what we like to know is that, do these strains exist, and can we then define them? Can we then use them to examine different cell type specificity, or different brain region selectivity, and also different rate of disease progression? And so the way that we do that is that, basically, and we need to study the human brain and the pathology in human brain before we can even model them. So the best way to really do that is to just really take human synucleinopathy brains. And so there are many different ones. Like, we can, as I mentioned, that Alzheimer's disease have a lot of alpha-synuclein pathology, a lot of Lewy bodies. So we basically take brains from Alzheimer's patient or patient with Parkinson disease dementia or dementia with Lewy body, and then we actually identify the region that have the most pathology, and then we take that brain region, and then we extract them, and I'll show you a little bit later the scheme that we do, and to generate these enriched alpha-synuclein pathology extracts, and then we inject them and directly into wild-type mice. And then we waited for a period of time, and then we look at the spread of the alpha-synuclein aggregates in the mouse brain, and then we can then compare the spread of alpha-synuclein aggregates between Lewy body strains and also preformed fibers. Remember that we started with preformed fibers, but now we want to do the real thing, look at the real thing, because preformed fibers are all generated from test tube, but we want to study the spread of alpha-synuclein aggregates isolated from Lewy bodies, isolated from human brain. So this is the cases that we use. So we basically use a number of Alzheimer's cases, a number of cases, and Parkinson's disease, dementia, and about 4 cases a day, 4 or 5 cases. And as you can see here, these cases actually have at other pathology, in 80 cases, there are a secondary neuropathological diagnosis of Lewy body disease. So in other words, they do have quite a bit of Lewy body in this Alzheimer's brain. And the converse is also true, too, for some of these PDD cases, and some of them have concomitant Alzheimer's disease as well. So we took these brain, and you can see the demographic of the brain. It just doesn't advance. No, I want to, oops, sorry. Sorry, this is not listening to me. I want to go backwards now, but it's not letting me go backwards. Yeah, if you can help me. Oh, yeah, all right. Go backwards. Backwards more. Couple more. More. Yeah, this is fine. Yeah, okay. So basically, and so we selected these brains, and then we basically characterized alpha-synuclein pathology, and that are found in AD and PDD brains. And this is just to show you that every single one of these cases and have alpha-synuclein, phosphorylated alpha-synuclein pathology, and from both AD and PDD brain. And so basically, we were able to, make this extract from these brains, and then we basically, the way to test whether or not they have the ability to induce pathology is to take this enriched extract and throw them into primary mouse hippocampal neuron, or mouse. We use hippocampal neurons, but you can also use other neurons as well. And then basically you can see that these different preparation of Lewy body, enriched Lewy body preparation, and from AD cases or also from PDD cases, develop an induced Lewy body at very low concentration at 20 nanogram. And the same concentration of PFF, we saw nothing. And so, so basically, and what we found in some of these experiments is that the brain-derived and Lewy body extracts are much more potent in inducing pathology than preformed fibrils. And so this is just high magnification to show you that they also induce neuritic pathology too, but they've actually, the most striking and things that we saw was this Lewy body inclusion, much like what you see in human brain. So then we want to know whether or not there's a dose-response relationship. So in other words, that is this material due to alpha-synuclein? If we increase the concentration of this Lewy body extract, do you see increased number of Lewy bodies? And indeed, that's true. So with low concentration, like 5 nanogram, you see a few Lewy bodies. But then if you look at higher concentration, like 30 microgram, or 30 nanogram of alpha-synuclein pathology, you can see that there are quite a few more Lewy body found in these neurons. All right, so you can see that these Lewy body extract at different concentration induce Lewy body pathology in primary hippocampal neurons. And so in contrast, if you take now and preformed fibrils, you can see that they don't really induce much Lewy bodies. And for example, you can see all of these are neuritic pathology, the green that you can see in this slide at the different concentration of preformed fibrils, even at the highest concentration, and induce more neuritic pathology and not Lewy bodies. So there's a distinction between the ability of preformed fibril to induce pathology versus from Lewy bodies. So this is a quantification just to show that in terms of cell body Lewy body-like pathology, all the brain extract Lewy body cases show large amount of somatic and pathology, but not the preformed fibril. The preformed fibril really have a lot of pathology, but not Lewy bodies. And so, but as you know, that it's all very nice that we can extract stuff from human brain and show that they induce Lewy body, but that material is limited, and not everyone have access to human brain samples, and in fact, part of the reason why we use Alzheimer cases and Parkinson cases is because of the fact that there's a lot more Lewy bodies. That's why Alzheimer cases we use that as a start material. But then, how can we make more of it so that we can actually share it with our colleagues? And not everybody have, as I said, have access to human brain. So what I came up with the idea is that can we sort of amplify the conformation, the abnormal conformation in these Lewy body extract from Alzheimer cases or from Parkinson's disease cases. So the way we do that is that we take about 5% of the seeds from the human cases, from either Alzheimer's or DLB Lewy body seeds, and then we add alpha-synuclein monomers, 95%. Basically, these are made in the laboratory, human alpha-synuclein made in the laboratory. And then we agitate it and for X number of days, and we call that amplified Lewy bodies. And passage 1, okay, because we amplify that with seeds. And then we take a little bit of the first generation, the P1, and then we shake them again or incubate whatever condition that you want to use with more monomer, alpha-synuclein monomer, and then we generate what we call P2. The idea is that can you passage the Lewy body and then retain the conformation that you find in the human brain? And so this is the way that we did it, and then I'll show you some of the data that we have, and then we test them over time. So first is that we ask whether or not, if we do this incubation, that they're all aggregated. So in other words, they would be palatable. So in other words, they become aggregates. They're not in the soluble fraction, but they're in the insoluble fraction. Indeed, that's the case. Even by 4 days, and the majority of the material are recovered in the palate fraction, you can see that all the different cases behave very similarly in the sense that they all are capable of inducing an aggregate. And so do these aggregates have the properties of inducing Lewy body, just like the start material? The answer is yes. So basically, we took the P1 seeding, and then we basically did that over time. And you can see it at 4 days, 7 days, 10 days, up until 21 days. Again, just like the initial seed material and the amplified P1 material induced Lewy body inclusion in a dose-dependent manner. And they're all, and both all the cases either from Alzheimer's or from Parkinson's. And so the induction of the pathology, all the cases, as you can see, and they're all induction of phosphorylated alpha-synuclein in the hippocampal neurons. That they're treated with the amplified Lewy body. And so this is just a quantification of the P1 and with human and AD and then DLB cases, and they all are capable of inducing Lewy bodies and the P1. And what about P2? Well, P2 is more difficult to do. So basically, we found that P2 also does the same thing. You can see that there's like, a couple of cases of P2 and at different concentration. You can see that they both induce Lewy body pathology. And so what are these Lewy body that are induced by P1 and P2 share the same property with the original material? This is just further characterization of the Lewy body. We show that they're the same in the sense that they're ubiquitinated, and they also have P62. Basically, the proteasome is trying to degrade this material, so therefore they coaggregate with with alpha-synuclein aggregate. So they have the same feature as those from the initial material that we isolate from brain. And so we want to know whether biochemically that they're the same. In other words, that if the conformation of the AD Lewy body or the PDD Lewy body are different, they would have different fragments when we try to degrade the Lewy body pathology with proteinase K. And so basically, this is what we do here. You can see that in all the panel with the AD cases on the top and PDD cases on the top, and then you can see that when we add proteinase K digestion, it generated 6 different fragments and in the bottom of the gel there. And it seemed like that, that the AD cases and the PDD cases, when we treated them with proteinase K, they're quite similar to each other in terms of the fragment that they generate. Some have more, some have less, but they're similar to each of them. But you can, if you look at that pattern, 6 fragments compared to the PFF, which is up there and on your left, and you can see that the PFF only generated 3 major fragments. So there are clearly differences in conformation between the preformed fibril and the AD and PD cases. So that may explain why they show different type of pathology, because conformationally, preformed fibrils are different from the brain-derived Lewy bodies. So finally, we want to see whether or not these amplified Lewy body can induce pathology, Lewy body pathology. And in animal model, at the end of the day, it's really desirable to have animal model that would actually and develop Lewy body with the amplified Lewy body. So here, what we did was that we actually took 10 times less an amplified Lewy body than human PFF. You can see that we can see pathology, and indeed similar to what we saw before. And in the amplified Lewy body, they induce more Lewy body-like structure and than human PFF. And the quantification on the side, you can see that they're by and large in the same region of the brain, but in the distribution in terms of intensity are different from each other. So to conclude this part of my talk, basically what I've shared with you today is that pathological alpha-synuclein from patient-derived Lewy bodies induce pathology that is distinct from that induced by preformed fibrils in our culture primary neurons. And furthermore, Lewy body alpha-synuclein, they're more potent than preformed fibril at seeding alpha-synuclein aggregation in hippocampal neurons. And that this Lewy body aggregates and can be amplified in vitro using recombinant alpha-synuclein, generated aggregates that maintain the unique pathological phenotypes of the original material from diseased brains. And amplified Lewy bodies induce alpha-synuclein pathology in several brain regions in wild-type mice, and the spread of pathology induced by amplified Lewy body are distinct from that of preformed fibrils. And then that's why we think that the Lewy body alpha-synuclein, they elicit a unique cellular response and not recapitulated, not exhibited by and recombinant, fully recombinant fibrils. And so, but the fact that we can amplify Lewy body and that we can share with the community will facilitate research in this area in Parkinson's disease and dementia with Lewy body and for the development of therapeutics. So I want to conclude by thanking the people who did the slide. Actually, these are just some of the key people, and a lot of people contribute to the study from our center. But Jahan Ara and Nick Murata were the one that actually generated the amplified Lewy body, and Norihito Umura is the person who did the animal model, and Kevin, Luke, and I started a lot of this Lewy body transmission together more than 10 years ago now. And Bin Zhang is the surgeon and who did the injection, and you need somebody who do the injection can do it well. And Lashmi is the champion because she's the one that make all of the extracts from human brain, and then all the study were done in collaboration with the late John Turjanowski. I thank you for your attention.